and I have known for a while, each other for a while. He was invited to the CIC seminar series in 2018, I think. Actually, you were my first invitee. So I mean, it's the opposite of the gospel. I get the first wine first. Alexis is a world-class researcher working in the field of actin regulation. I mean, his lab studied the cellular regulation of the ARP2/3 complex, which, as you know from yesterday's lecture and you will learn today, it generates actin branch networks. He did his postdoc with Marat Kirchner, who's a big name in Harvard Medical School, where he purified the architecture and characterized the architecture of the wave complex, which generates branch actin networks in membrane protrusions and mammalian cells. Back in Paris, he discovered the analogous wash complex that controls actin dynamics around endosomes. And more recently, he discovered a negative regulator of the ARP2/3 complex named Arpin, which antagonizes the wave complex at the leading edge of migrating cells and that controls migration. After that, he became, or maybe at the same time, he became interested in cancer, and he discovered that tumor cells lose expression of this migration inhibitor, and this is heavily involved in metastasis. I hope he will tell us a bit about that. And well, I mean, he leads a really exciting lab which combines like clinical data with high-end biochemistry, like old school biochemistry, to dissect the roles of molecular mechanisms of the ARP2/3 regulation in normal tumor cells. I mean, so you know the type of person you are dealing with, and now he's blushing a bit, but I have to do these things. He's a research director at the CNRS, which is the French equivalent to CSIC. He's a full professor at the École Polytechnique, which is the #1 engineering school in France. And he's a visiting professor at the Moscow Institute of Physics, where he's currently, as some of you could hear before, he's setting up a second lab, which is gonna steer his lab in very new and exciting direction. So, just to thank you again for accepting our invitation, Alexis, and well, the floor is yours.
Okay, thank you very much, Miguel. So, for the students, I don't see you. I believe you are not so many, so please interrupt me at any time. This lecture is really for you, so don't hesitate to interrupt. But this being said, I would like to tell you that I'm recording this lecture because I plan to use it for other online courses. So don't be shy, it's not a problem, but don't hesitate to interrupt. So let me share my screen now, and okay, so I guess you can see my slide.
Yes.
Can you?
Yeah.
Okay, I need a pointer, that's fine. And I need to start it. Well, wait. I lost my pointer.
We can see your arrow fine anyways. I mean, so whatever it's more comfortable for you.
Do you see the red point now moving?
Both of them, yeah.
All right, so now only the red point should be here.
Mm-hmm.
All right, so I will tell you about branched actin. So Miguel told me you had an introductory course on the actin cytoskeleton, so I will directly take it from here, branched actin specifically, and we'll talk about its role in cell migration and cancer progression. And as Miguel said, I worked in École Polytechnique, so just a couple of words about Polytechnique. This is an old engineering school in France. This is a top one in France. It was created right after the French Revolution in 1794, and it became a military school by the will of Napoleon because it was very important for the country to build bridges, to build weapons and all this, and hence it explains this a bit out of fashion symbol, which is, you see, a military shield, and the motto is for the Homeland Sciences and Glory. So, of course, it's a modern school now. Actually, this concept of engineering school called Polytechnique really spread across the world. And we are very proud that this school was the first one to be created. So we are working in the south of Paris, in Palaiseau, in a small city called Palaiseau. And so I must tell you that the invitation that Miguel referred to was to celebrate the anniversary of 800 years of the University of Salamanca. So I feel ridiculous when I say École Polytechnique is an old institution. It's not that old by this standard, not at all. All right, so today I divided my talk in 3 parts. The first part is really textbook knowledge about the ARP2/3 complex and its activation. Here, I will try to show you where the most important knowledge we have about the ARP2/3 complex comes from, how it was demonstrated for the last, let's say, 25 years. And then I will focus on 2 aspects that my lab is developing. One is about the study of an ARP2/3 inhibitory protein that we call the Arpin. And the last part is about a new unexpected function of branched actin, which is to control cell cycle progression, so proliferation of cells. And so we'll have a break in the middle, so in the middle of the second part. And all that is related to cancer progression will be after the break in the second part. All right. So a basic introduction. You know that for migration, cells become polarized. So in this kind here, you have a scanning EM of a migrating cell, and it's characterized by a large front containing a protrusion of membranes like lamellipodia, one lamellipodium, several lamellipodia. We'll talk about that extensively, or finger-like protrusions called filopodia. And so you see that the front is quite large, whereas the rear of the cell is kind of tiny because it's under contraction. Here, I just showed you by transmission EM this time, the ultrastructure of these membrane protrusions. So you kind of see some filaments in lamellipodia, but this is a very dense meshwork, so you hardly see their organization, whereas for filopodia, you get a sense that within the lamellipodia with a dense meshwork, you have linear bundles of actin filaments that are really below and pushing the membrane in these protrusions. And in a way, you know, the actin cytoskeleton is responsible for all kinds of shapes in the body. So here I showed you the microvilli of the absorptive gut, and you have the same kind of linear arrays of actin filaments, actin bundles, to increase the surface of exchanges between the extracellular milieu and the cell. So, as I said, we will focus on lamellipodia a lot because it was discovered by Tatiana Svitkina in 1999 that this dense meshwork is actually composed of branched actin. So Svitkina was a famous microscopist, and she had developed a method called platinum replica. So you fix, you deterge cells, you make a replica, and you observe by EM. And these numerous branches that you can see, for example, here, of actin filament, raised a lot of criticism at the time because people thought that they knew perfectly well how actin was polymerized. It was a long time that actin, which is very abundant, could be purified. Spontaneously in the test tube, it polymerizes, and no one had ever seen branched filament. It was always linear filament that were obtained in this case. So this discovery was welcomed with a lot of criticism, actually. So a very important message is that the actin cytoskeleton, which is responsible for the overall cell shape, is responsible for the overall cell shape because it can do many things. And in particular, it generates forces. It generates pushing forces. And this is the role of actin polymerization alone. It does so in protrusions like lamellipodia, where we've seen filaments are branched, or filopodia microvilli, where filaments are not branched. But it can also pull, and we will not discuss this further in the talk, but this is an essential role of myosin 2, and I'm sure you will hear about it from Miguel. And here I just put the different structures where actin is contractile. It can be the contractile ring when cells divide. It can be the adhesion belt of epithelial monolayer. It can be the stress fibers when cells spread and pull on the extracellular matrix. And yeah. All right, so lamellipodia. A very important basic knowledge you should have is that, so you have a lot of actin in the width of lamellipodia. In this movie, we performed, when I say we, sorry, I borrowed this data from the group of Klommensrotner. They did photo bleach this central part of the lamellipodium. And actually what you can see is that the recovery starts from the membrane and then extend backwards. And this is the so-called retrograde flow of actin. Actin is polymerized right beneath the membrane and then is pushed behind by the new actin polymerizing at the front. So this is a very important conclusion that one has to keep in mind. And of course, just looking at GFP actin, you see the steady state. So you see this whole width of lamellipodia, but you need to induce a perturbation like a frap to see the dynamic within this structure. So many people were excited about the mechanisms of cell migration because it's so important for, let's say, embryonic development or immune system with all these cells that are patrolling your body, or let's say metastasis, and we'll talk about that later. But really, it was a very hard topic because even though actin is an abundant protein, I mean, if you analyze migrating cells, actin is polymerized at the very front of the cells, is under contraction at the rear, which is tiny in this tail that I showed you, and is depolymerizing over there. So everything is like mixed up. And the real progress came through a detour by this intracellular bacterium called Listeria. Here you have a cell which is infected by Listeria, and when you label GFP actin, you see these amazing comet tails that are propelling intracellular bacteria. At the tip of this tail, you would have a single bacterium in each case. And this was understood pretty early that it would be a very useful system to decipher the mechanisms of actin polymerization. So one breakthrough in the field was, of course, to identify the membrane protein from the bacterium. It's called Actin, that was responsible for these actin comet tails. You see that here the bacteria, the Listeria, are propelling themselves thanks to actin comet tails. In this mutant, the intracellular bacteria can proliferate within the cell, but they cannot propel themselves. And actually, this propelling is very important for the bacteria to infect neighboring cells from the first infected cells, and so to spread the infection without ever becoming extracellular. So without being seen by the antibody of the immunity. And so, this discovery by the group of Pascal Cossard in Institut Pasteur in Paris was really the beginning of the dissection of the mechanism I will tell you about. The group of Matt Welch, you see, purified the protein and covered inert beads, polystyrene beads, if I recall properly. And basically, in a cytosolic extract, he managed to reconstitute the movement of inert beads in a soup containing all proteins of the cytosol. And so here you see labeled actin, fluorescent actin, and you see movement is reconstituted with no living cell, just in an extract. And this was actually very important to make this demonstration because Matt Welch was the one who understood the role of the Optus3 complex. And so at this point, I like to ask my students, okay, imagine you are exactly here at this point, you know Actin, so you have a molecular handle to dissect the mechanism. What would you do with Actin to try to dissect these mechanisms? So please suggest what you would do with Actin in hand to identify mechanisms. If you unmute your mic, if you get ideas, what do you suggest?
If you want to answer, just raise your hand, and I'll give you the microphone.
They cannot take it right away just by pressing the space bar or something.
I think I have to give them permission. I think because they are participants, not panelists.
Okay. So give them permission if you can, and
Sure.
For the next time we'll see if it works better, more spontaneously. So I will give you the answer. The classical answer, it's a trap, because most people say, okay, I've got Actin, let's see to what Actin binds. And you can use all kinds of techniques like, I don't know, GST pull down, or all kind of affinity chromatography, or histohybrid screen to try to find the proteins of the host cell that will bind to the bacterial protein Actin. And actually many labs around the world tried to do that, and success was very, very limited. And I can talk about it because this was what I was given when I was a master's student as a project in Pascal Cossard's lab. And so I was supposed to try to perform a histohybrid screen, and it really went nowhere. And actually Matt Welch cracked the problem. And the way he did it was to use in vitro reconstitution. So what is in vitro reconstitution? This is based on the old school biochemistry that Miguel was referring to, but it's actually very elaborate. What you want to do is to reconstitute an elaborate cell function in the test tube. And they were very close to doing that, because they knew that with a cytosolic extract, they can reconstitute the movement of beads or of listeria, and that you can follow the process just by doping in the extract fluorescently labeled actin. And so this is what you see indeed in this soup of proteins. Bacteria do move. But Matt Welch used this assay to fractionate the extracts. So you have many techniques available to do that. Let's say ion exchange chromatography, gel filtration, ammonium sulfate cuts, so on and so forth. You may not be familiar with these techniques, but this is really the old school biochemistry. You have a complex mixture of proteins, and you will separate proteins in different fractions. And basically what he figured was that some fraction, a few fractions were active, and most of the others were not. So with the active fraction, you can reconstitute some actin polymerization around the bacteria. It never develops fully into a comet tail because we will see that it requires other components, but you had the basic actin polymerization. And so by several steps of fractionation, Matt Welch figured in this last step, which was a gel filtration, which separates proteins according to their size, or complexes according to their mass, to their size, shape, that you had 7 proteins that were co-eluting in the same fractions. So that's a very strong evidence that it is a multi-protein complex. And he could identify the identity of this complex by mass spec. And this complex was the ARP2/3 complex. It contains 2 actin-related proteins, ARP2 and ARP3. This complex was discovered by Laura Maciejewski when she was a PhD student with Tom Pollard. Those are all big names of the field. And, but really, this experiment was the beginning of the understanding of the role of the ARP2/3 complex. And so once, okay, so the beauty of this in vitro reconstitution and this purification based on activity is that you're sure not to be misled. You're sure that you will find significant things. So this thing should play an important role in actin polymerization, which is not at all the case, for example, for some things that randomly binds to Actin, as you can find by histohybrid screen or by all kind of affinity chromatography. So it was a very direct method. So the old school biochemistry is very important to know, because it can be applied in some cases, and this is how you make really a great progress. So after having found the ARP2/3 complex, they wanted to validate that it was significant. So they raised antibody against ARP3, and then you label comet tails. And one of the surprise is that the whole actin comet tail was labeled. Not only, you would expect it only, if it was playing a role purely in actin polymerization, you would expect it behind the bacterium at the interface between the comet tail and the bacterium, but it was throughout the actin comet tail, just like actin itself, okay? And so I'll try to see if you can participate now. So this was done in 1997. There's one expected validation that one can think of, which was not the case at the time. What kind of validation, what kind of additional validation can you suggest? What would you do now to be sure that the ARP2/3 complex was involved in the propelling of this bacterium? Go ahead, I could hear some.
Knockout, maybe?
Exactly. You would do a loss of function experiment. It can be a knockout by CRISPR. It can be a knockdown by siRNA. It was not existing at the time. It was a pre-RNAi time. So it was not in the paper, but this is what we would do now for sure. All right. So Matt-Welch, after...
Let me just say, all of you are now enabled to speak, but I would ask that you keep your microphones shut until you want to speak. Then you open it and then you close it, so we eliminate some of the background noise and we can have a very clear audio, which we are having. Sorry, go ahead.
Yeah, yes, okay. So there was another paper published in Science just 1 year later, where the core of this paper is this figure, a very simple figure. This is a spectropluorimetric assay. So basically you monitor in real time actin polymerization in the test tube just by following the fluorescence of this compound. So you purify actin, you label it with this fluorophore, which is pyrene, at a specific position on actin. And basically when actin will be part of a filament, this compound will fluoresce much more than when it is free globular in solution. And so you can monitor spontaneous actin polymerization. This is a spontaneous actin polymerization. Unfortunately, the curve was not on the figure. So this is actin alone. ACTE that you add to it does nothing to it. If you add purified ARP2/3, you see a little bit of enhancement in the speed at which actin polymerizes. But if you add actin, the ARP2/3 and ACTE, boom, you get a super fast actin polymerization. You see the slope here. This is the slope that matters. And so ACTE was the first activator of the ARP2/3 complex that was discovered. All right. And so we could cover beads with ACTE. We could make it work with purified ARP2/3 and actin. And you get the exact same branches that Tatiana Svitkina had first reported in the cell. So this was no artifact. This was a product of the ARP2/3 complex. I would like to mention this study because it was quite important in the field. So this is a Nature paper in 1999, produced by the group of Marie-France Carlier. Basically, she went ahead and went one step further. What they did, they knew that you could reconstitute the movement of Listeria in cytosolic extracts. And they said, I want to understand exactly what is the machinery responsible for the movement. So I want to reconstitute it with pure proteins. What Matt-Welch had done was to reconstitute the initial actin polymerization using actin and ARP2/3. And she found out with her group that she needed to add a capping protein which terminates actin polymerization and to add cofilin that depolymerizes actin in order to sustain the movement. You see, in order to depolymerize the tail after it was produced so that the movement could be sustained. And this was really a tour de force because as you can see on this graph here, the amount of each protein needs to be very precise. You need to add enough for the movement to take place, but not too much. Otherwise, you would imbalance the reaction toward polymerization or depolymerization or termination of actin elongation. and so movement would not be sustained. So it shows that actually to make this kind of reconstitution, you need to have a very accurate knowledge of what the different actin binding proteins are doing, and you need to know at which concentration to put each of them in order to reconstitute the movement. So that was really a breakthrough in the field. And so what we understand now for sustained movement, and this applies for listeria that would be protruding here, or for lamellipodium, which would be this membrane here, is that the ARP2/3 complex that is this green guy here creating branches is multiplying the number of filaments and the number of ends of filaments pushing against the membrane. And in order to have productive force generation, you need to terminate filaments which are not pushing against the membrane. And in order to renew the pool of polymerizable actin, you need to sever the old filament using cophylline. And this is how movement is sustained. So as for the suggestion, indeed, so in 2006, it was post-RNAi discovery, and the group of Clemens Rotner could inactivate 2 subunits of the ARP2/3 complex. And you can see here on these cells, these melanoma cells, making all these lamellipodia. Those are all these concave protrusions that are missing here, that the ARP2/3 complex was required for lamellipodia formation. Here, another example coming from the group of Rong Li. She used a mouse embryonic fibroblast derived from the ARP-C3 inactivated mouse. And basically, you see a single lamellipodium extending throughout the periphery of these cells when cells spread on the substratum. I will play the movie again. And you see that you have a very smooth spreading, which is not at all the case in the knockout maps, where you see, I will play it once more, you see filopodia are still being produced with no problem because it requires linear actin and no branched actin. And basically these filopodia get thicker and make this kind of stubby protrusions that will eventually spread the cell, but the membrane in between filopodia cannot be projected. The role of lamellipodia would be to project lamellipodia in between these stubby filopodial protrusions. Okay, and so this is, and so the result in terms of morphology is completely different. All right, so remember that the actin cytoskeleton pushes, and this is a role of the ARP2/3, and pulls, and this is a role of myosin. And we know that for sure, thanks to in vitro reconstitution, because we know that we can reconstitute the force in vitro and that we don't need myosin in there. So pure actin polymerization is generating the force. Now, a bit more about the ARP2/3 complex. If you label one subunit of the ARP2/3 complex, you find it where you expect it. So here it's a mouse embryonic fiber, well, it's 3T3. So you find it in the lamellipodium, in the width of the lamellipodium, just like actin. But you see also numerous intracellular organelles. And actually, the role of the ARP2/3 is very important for organelle. This is not something that I will develop in this lecture, but this is equally important. So when the ARP2/3 was purified, its crystal structure was cracked by Robert Robinson and the group of Tom Polla. And it has this kind of architecture where the 2 actin-related proteins, ARP2 and ARP3, are quite apart. This is an inactive conformation. And if you use an activator like Actin or some endogenous activators that the cell produces that we will call from now on nucleation promoting factors, or NPFs, then you induce a conformational change that brings together ARP2 and ARP3, and this rearranged ARP2/3 has affinity for a pre-existing actin filament. So this is why it lands on a pre-existing filament and induce the formation of a branch, because these 2 ARP2 and ARP3 look like the end of actin when they are in close proximity, and so it can induce a branch. So it's regulated at several levels, and we'll come back to this. But let's focus first on the most important aspect, which is the activation of the OP2/3 complex. So by a lot of techniques, some proteins were found to bind to the OP2/3, and they were afterwards tested in vitro. And these proteins activate in the test tube the OP2/3 complex, and they are characterized by very similar C-terminal ends containing two motifs: the acidic motif and one or several WH2 motifs. And the two motifs are absolutely required for activation of the OP2/3. So when I say motif, it's really, it's a short linear segment of protein. It's not a folded domain. It's not a domain, it's a motif. The acidic motif will induce this conformational change I was telling you about. And the W stands for WH2 motif, which binds to actin. And actually, it's absolutely required to deliver directly the first actin through a WH2 in order to create this structure, which is the first structure, this trimer, that really looks like an actin filament, so which can then be elongated. If you don't have a WH2, the daughter filament does not get extended. And so these four classes of activators have distinct function. The role in lamellipodium protrusion is really the role of wave, and we'll see that in detail. And for NWASP, WAM, and WASH, these three other family of proteins are involved in intracellular trafficking at different places during endocytosis, ER to Golgi transport, or at the level of endosomes. So you have a division of labor, meaning that even though you have branched actin networks at many different places of the cell, at a first approximation, they are nucleated by different activators. And so that makes our life simple to decipher the role of the OP2/3 complex. So just to illustrate this point, even though it's not going to be developed, you have here a clathrin-coated pit. So this is a process of endocytosis. You have a series of bar proteins which bind membranes with different, which induce curvature. And so they will polymerize around the neck of the endocytic pit, but they do not suffice in most cases to induce a scission of this pit. And this is the role of branched actin polymerization to really contract and compress the neck to help scission. Even though the architecture of the branched actin network is not yet deciphered, we found actually that the WASH complex may play an homologous role of in scissioning transport intermediates from endosomes where sorted cargo are accumulating. But of course, for this lecture, we'll focus on wave. And here I took this, this movie from an old work from Penelope Hanna. What you see here is that wave labels very distinctly the very edge of lamellipodia. And not only this, but you will see that here in the center, you have a retraction in the middle of the movie. And when the membrane retracts, the protein is gone almost instantly. So in other words, you have a perfect correlation between the protrusion on the membrane and the presence of wave, which is a pretty good evidence that it is involved in protrusion. Of course, this was confirmed by the expected loss of function experiments, knockdown, knockout, and wave proteins are really critical for lamellipodial protrusions. So wave itself is contained within a multi-protein complex, just like the OP2/3 complex. So this is a cascade of complex working together. We know the overall architecture. We have a crystal structure thanks to the work of Mike Rosen. And actually, the WA motif is masked by an intramolecular interaction within the complex. So this complex is initially inactive, and it will become active. So it will expose the WA motif to activate the OP2/3 in response to signaling. And the major switch in this response is a small GTPase RAC. So I suppose you're familiar with RAC. This is a small GTPase that belongs to the superfamily of RAS, but you know that you have a subfamily called the Rho family that contains the canonical members, Rho, RAC1, CDC42, which exert a major role on actin polymerization and actin organization in the cell. And RAC is a master switch to create lamellipodia because it's a master switch triggering branched actin polymerization at the lamellipodium edge. And the way to show that, I borrowed this very clear example from the group of Clemens Rottner, again, Annika Stephan and Clemens Rottner. Here they produced an active form of RAC1, which thanks to a mutation is always bound to the GTP nucleotide. And they will microinject cells. And in control cells, if you microinject RAC, you see a single lamellipodium extending throughout the periphery, just like in spreading cells, as I showed you. But if you deplete the wave complex now, or the ARP23 complex, but let's take the wave complex as it is depicted here, when you microinject RAC, you see that you don't have this spreading effect at all. And this is due to a defect in actin polymerization. Instead, you would have blebbing in some cases. And okay, I might comment on this in response to questions, but so this really shows that the wave complex is downstream RAC. So we can order this pathway, which is very clear. The wave activation of the ARP23 was reconstituted in vitro and was responsible for branched actin. And thanks to this kind of experiment, RAC was shown in cells to activate the wave complex. RAC binds to wave, RAC activates wave, and this was eventually reconstituted in vitro, also by the group of Mike Rosen. So this pathway is absolutely clear. That's not so often, so that should be emphasized. So remember that the ARP23 activation occurs at many different locations within the cell. And this is, for the most part, mediated by different nucleation promoting factors. And that in all cases, it generates pushing forces. We will discuss in details here the case of membrane protrusion in lamellipodia, but it can also compress membranes to remodel them to induce scission of transport intermediates at different places of the cell. So I go back to this scheme of the activation of the ARP23 complex. And so this is a biochemical equilibrium. So spontaneously, the ARP23 can reach this conformation, but it does so very infrequently. So this is activated by NPFs. But you also have some proteins whose role in the cell is to mediate the reverse reaction, is to favor the reverse reaction, which is spontaneous again. But kinetically, it favors it. And this is a role of Arpin, a protein we found and that we will describe now. So Arpin was found through a bioinformatics screen for containing an acidic motif at its very C terminus, but lacking a WH2 domain. Since I told you the WH2 domain is critical for actin polymerization, this thing cannot activate actin polymerization, even though it binds to the ARP23. So I will show you that it's an inhibitor, and we had to give it a name because really that was fully uncharacterized, and we called it Arpin. And this was a tribute to my PhD supervisor, whose name was Monique Arpin, Arpin in French, Arpin. And this was a predestined name because it was perfect fit for ARP23 inhibition. And so Arpin is this protein. So just to tell you, this protein was found by my first PhD student, Emmanuel de Rivery, who has now his own lab and still working on this topic. And it was a weekend work. It was no work on the weekend. He just decided to say, oh, perhaps there are more NPFs to find. And so he retrieved on his personal PC the UniProt database. He discarded all protein that did not contain a tryptophan, which is absolutely conserved at this antepenultimate position. and then rank the remaining proteins by a score of acidity. So the number of DE, very simple. That took him 10 minutes to program. And he found in between the NPF, the known NPF, he found this uncharacterized protein that turned out to be Arpin. So just to tell you that sometimes it's good to wander around and to ask some questions like this. So even though he wasn't the one to study it afterwards, that gave a Nature paper a couple of years later. So we were all grateful for this weekend of programming. So I borrowed this experiment from our collaborator, Laurent Blanchon in Grenoble, because this is turf imaging of actin polymerization. The complete reaction, so actin ARP23 WA. So you will see in real time formation of branched actin. So you look at actin, so you see the actin filaments, and you see branch creation, as simple as this. And this is, of course, fully ARP23 dependent. If you add Arpin on top of it, then you see much less branches created. So Arpin is indeed an inhibitor. And the way it works is that since it has an acidic motif, it's a competitive inhibitor for the NPF. It's a competitive inhibitor for ARP23 activation. So far, so good, but what is the role of Arpin in the cell? So actually, we looked where it was. We raised antibody, and that was the work of Roman Gorelik in the lab, postdoc. And we had the surprise to localize a protein at the tip of lamellipodia, so exactly where actin is polymerized. So we localized the inhibitor where actin is polymerized. That was a bit surprising initially, to say the least. That puzzled us for some time, but actually it makes a lot of sense, and we'll see how. So Irene Dang, PhD student, performed an immunoprecipitation of Arpin to see whether it binds the ARP23 complex. In all cells that are regularly living, making lamellipodia at some places, retracting it at some other places, you can see some level of co-IP of Arpin with the ARP23. But then we use mouse embryonic fibroblasts from Clemens Rotner, who were devoid of RAC1. And as I told you, RAC1 is a master switch for lamellipodia formation. So in these knockout maps, we've never seen a single lamellipodium, right? So these cells still express Arpin and the ARP23, but you see Arpin does not bind to the ARP23 if cells don't have lamellipodia, if cells don't have RAC. And that was the clue, because basically now we can draw this relationship like this. So RAC is activating the activator to trigger branched actin formation, but RAC is also activating the inhibitor to inhibit the ARP23 complex. It doesn't do so directly as it does for the wave complex. We have some intermediates here that we are still chasing. We don't know their identity, but RAC does not directly bind to Arpin. But Arpin, in order to inhibit the ARP23 complex, fully depends on RAC. And just to raise your taste about this experiment, which is absolutely beautiful according to the standards of signaling. So why is it beautiful? Because here you have an endogenous co-immunoprecipitation. You didn't overexpress proteins. If you overexpress proteins, you can make them bind even though they would not, they were not supposed to bind in this condition. Here, it was the endogenous Arpin binding to the endogenous ARP23. It would have been difficult to overexpress the ARP23 by anyway, because it's a multi-protein complex. And then, instead of triggering the signaling by overexpressing, let's say, an active version of RAC1, as most people do, and as we did initially, then we remove one signaling intermediate. So we modulate an endogenous co-immunoprecipitation by removing one component. So we have no artifact for sure of overexpression in this kind of experiment. If you just have experiments with overexpression, you will find that many of your colleagues and reviewers of your papers will ask you, please show us that it also exists at the endogenous level. So we had that in this case, everything is endogenous. So we are pretty sure about this result, but then what does it mean really? So as I said, we were puzzled for quite some time, and then we did as we should, that is to read. And so in addition to feedback that most of you know, so feedback is something that would detect something below, I mean downstream, and connecting on something upstream. Here it's not a feedback, it's a feed forward. You have two pathways to go from rack on top of the switch to branched actin downstream. You have the activatory pathway and you have the inhibitory pathway. So when you have two pathways, it's called a feed forward. And when the orders are antagonistic, activatory on the one end and inhibitory on the other end, it's called incoherent. And actually, Uri Alon had studied this kind of circuits in general and in theory. In the case of transcription factors also, it was very well deciphered. And basically what we understood thanks to him, to his work, was that these kind of pathways are very good to convert one off-on switch into a bell-shaped curve. How come? Why so? You would have a direct transmission to the off-on switch to wave because it's direct, it's fast, it's efficient. For Arpin, we know it's indirect. So the activation of the inhibitor will build up over time. And if you sum up the two signals, then you would have an initial activation followed by an inhibition. So this is a theory. This is what incoherent feed-forward loops are good at, okay? So do we have evidence that it really works this way? So if you want to highlight the role of an inhibitor, you should start from a very efficient system of cell migration first. And the most efficient vertebrate cell system for cell migration is the fish keratocyte. So this is an experiment we've done with Vic Small in collaboration. Those cells come from fresh trouts from the market. So it's not a genetic system. You buy fresh trouts, you pull scales from the trout, and you will take the epithelium that covers the scale. And this is an epithelium which is very plastic, which is very good at repairing wounds. And if you dissociate the cells of this epithelium, you have exactly this. You have the fastest migrating cells ever. You can see the counter here. They migrate of their diameter in 1 minute or so. Those are the only cells from vertebrate that you can see moving with no acceleration, just under the microscope, you see them moving. And not only they do move, but they move in a relatively straight manner by having this shape with a gigantic lamellipodium covering half of their periphery. And they do move without changing shape. So this is a pretty amazing system. It has fascinated physicists, modelers, many people. So, but it's not a genetic system. So we cannot do a knockout, knock-in, overexpression. We cannot do this kind of things. So we went for the old school biochemistry. We cloned zebrafish Arpin. We purified it from bacteria or delta A, the same Arpin molecule lacking the short C-terminal motif. And we micro-injected these proteins into fish keratocytes, or just the buffer that contained the protein. So you can see if you inject the buffer or if you inject the delta A Arpin, not much happens. After the micro-injection, cells are a bit troubled, but they manage to continue their directional migration. The story is completely different if you put full-length Arpin in this system. You see that after micro-injection, cells start to change shape tremendously. So they still form lamellipodia, and this is the first very important conclusion. If you put loads of Arpin in cells, you cannot block completely lamellipodia from being emitted, but instead you're rendering them very unstable. So they are protruding, retracting, protruding, retracting. And as a result, of course, cells do not migrate as efficiently. They lose their polarity, so they change direction when eventually they can resume migration. And so you mess up completely with migration and you render it inefficient. So how can we understand this in the scheme of cell migration? So it was known through the work of modelers that to sustain movement of a fish keratocyte, you need positive feedback loop. And positive feedback loops, we know there's one that detects where actin is polymerized at the front of the cell, and that will, as a result, activate RAC downstream. And actually, this pathway was worked out by the group of José Bustelo in Salamanca. It's Antonio Castro and Bustelo showed that coronin detects branched actin and associate with a RAC exchange factor that will continue to activate RAC wherever it was previously active. So you sustain actin polymerization, so you sustain the front at the front, and so you sustain movement and you make a very directional migration thanks to this positive feedback loop. Now, what does Arpin do in this context? This is the same movie as I showed you previously. This is the one where we injected Arpin. But what we did here is to change reference. Instead of looking at the cell moving as everyone does, meaning you're in the framework, you're in the reference framework of your microscope, we will try to be like observing the situation from the cell point of view. So for this, we draw the outline of the cell and we calculate the center of the cell. And we will translate frames of the movie frame by frame so that the center of the cell remains the center. This is a change of reference. This is as if you were observing the scene from the cell's point of view. So what we've done here is to remove all movement of the overall cell. And we needed that to make a kymograph of what's going on for the protrusion. And as you can see, if you make a kymograph almost anywhere in the cell, you see beautiful periodic oscillation of protrusion, retractions, protrusion, retractions, protrusion, retractions, and so on. And only when you put Arpin. So can we understand why you have this oscillatory behavior? Yes, actually, it's fully explained by our scheme. So you have this positive feedback loop that makes migration persistent. And what we have now is loads of Arpin that will favor this inhibitory pathway, this inhibitory arm that creates an incoherent feed-forward loop, so that is a bell-shaped response. So you continuously activate RAC, but you have this scheme here that make it bell-shaped. So you have the protrusion retraction. That's end waving, but this is what we believe is happening in this case. And you will see that actually Arpin is playing exactly this role. I should say as well that in this field, in this signal transduction pathway from RAC to branched actin, you have numerous intertwined feed-forward and feedback signaling. This was a Bustelo feedback, positive feedback, coronin 1A beta peaks, the RAC exchange factor that continued to activate RAC where it was activated. And this is our polymerization pathway. We have the Arpin inhibitory pathway. So you see all these things are intertwined. And basically the take-home message from this is that it's not interesting anymore to study signaling if you don't put time resolution into the analysis. Because basically what all this feedback and feed-forward will do is to regulate the temporal responses and so to regulate persistence of migration, which is the most important thing. So what was a surprise initially is that ARP2/3 inhibition appears to be a local event and it's not a way just to remove any noise from the system globally in the cytosol. No, it's locally regulated. And in the case of Arpin, this is to regulate migration persistence. All right, so I think I'm on time. We take a break of 10 minutes, so we will resume, or perhaps a bit less, at 9:30. And the second part is about the role of everything I told you in cancer. Okay, thank you. So I put the recording on hold.
Okay, so we'll see you in 5 minutes.
So it should be okay. All right. So second part. Second part. So we'll talk more about cancer progression in this case. And my model is mostly breast cancer. And actually, cell migration is very important for metastasis formation. Of course, cells are transported far away from the primary tumor site by streams like the bloodstream or lymph vessels, but they need to reach these vessels. So they need to crawl through their environment to reach the vessels, and then at distant places they need to so-called extravasate. So they need to exit these vessels to seed secondary tumors, liver in this case. In the case of breast cancer, liver is a very classical place where mammary tumor cells make metastasis. So when we started to study cancer, we took like a model of invasive breast mammary carcinoma line, which is MDA MB231, a very famous cell line. And we were surprised after the fish keratocyte experiments how inefficient these cells were. If I play this movie, you will see that the control cells are making protrusions absolutely fine, but they're not translocating, they're not moving anywhere. They stay pretty much where they are, despite their activity, their protruding activity. And when we depleted, thanks to RNAi, Arpin, you can see that now cells are really moving to different places. They're translocating themselves elsewhere. But as I said, this movie is a one-day movie, 24 hours. And so to see a little bit of migration, you really need to record on a stable system where cells are every 10 minutes, I think it was in this case. The same is true in 3D. So if we embed the same cells in collagen gels, you see the protrusive activity. So those are not flat lamellipodia, those are projections which are not as flat, but they are still driven by the RAC wave OP2-3 pathway. But again, and this is now a 2-day movie, again, cells don't move anywhere in the collagen gel. If you remove the Arpin molecule, this OP2-3 inhibitor, then you can follow almost any cell of this movie. You see that cells are really moving thanks to protrusion. And so we applied to trajectories many analyses. One is mean square displacement. So this is an area explored at different time intervals in movies. And sure enough, in 2D or 3D, the clones that were devoid of Arpin explored a wider area in these random cell migration assays than the control. So Arpin is globally an inhibitor of cell movement and cell translocation. And if we looked at patient samples, and we did that with Antonina Alexandrova in Moscow, so I show you, just for clarity initially, the two extreme cases. So a normal duct, so you see here the gland part, the duct part, which is composed of two epithelial layers, keratin 8 for the luminal layer, and keratin 17 for the myoepithelial layer surrounding it. These two epithelial layers do express Arpin, much more than the mesenchyme around it, which is a fibroblast and many adipocytes in the human breast. And this is consistent with the fact that these epithelial cells are going nowhere. Here you have one example of an invasive carcinoma. So you see these cells which are all keratin 8, so these luminal cells which have invaded the breast. And even though they were epithelial to start with, these cells do no longer express Arpin. And we, of course, have many such cases. So we also collaborated with Yvan Bièche in Institut Curie in Paris. And what Yvan has done is a retrospective cohort of patients afflicted with breast cancer that he followed for 30 years. 360 months is 30 years. And so we can measure the level of Arpin expression at the mRNA level by qRT-PCR. And we found 7% of the patients having a low level of Arpin expression, severe downregulation. And as you can see, the prognosis in terms of survival is poorer than the rest of the cohort. And so this is a P associated with such a distribution. So this is significant with 0.05 is a standard for this kind of clinical assays. So this is significant, but it's still not a very significant P. So let me explain what we draw here precisely in this Kaplan-Meier representation of survival. Here, this is metastasis-free survival. So it means once you have diagnosed breast cancer, by surgery, you remove the primary tumor before any treatment. And this is where you will assay Arpin expression. And then you let the system evolve, you let the patients live, and some of them will die from their cancer. Some of them will die from any other causes. They just cross the streets without watching. Or some of them will just move to different cities and we lose track of them. So those are all reasons to exit the cohort, right? And metastasis-free survival, you have one additional criterion, which is metastases are detected in this patient. Once you detect metastasis, the patient exits the cohort. So, and this is important in such a long retrospective cohort to exclude patients once they have metastasis, because over 30 years, the treatment has changed tremendously. So we cannot compare patients anymore, but we can compare the time during which they survived without metastasis. So with this criteria, metastasis-free survival, we say, okay, Arpin is not enough. We know how that works. We know Arpin antagonizes the wave complex, so we should measure expression of the wave complex. Of course, it's more difficult because you have 5 subunits of the wave complex, and each of these subunits can be encoded by paralogous genes except BRIC1. And so we measured all 11 genes, and actually NCKP1 we knew was the master because the level of its expression determines how many wave complexes you will find in a cell. And we found that this was a factor which was most strongly associated with prognosis. And this is how the survival curve, the metastasis-free survival curve looks like. You had overexpression in this case and not downregulation as found in Arpin in 40% of the patients, and the P was better, 10⁻³ in this case. But actually, to have migration, you can either overexpress the activator or downregulate the inhibitor. And if you combine the two criteria, you really divide your cohort into almost two equal parts, and you see how the P is becoming extremely, extremely significant. So we have a very good prognosis tool just by looking at 2 genes. So these results were patented and published. And of course, it makes sense according to our scheme that we got the same result, which is to induce motility if you overexpress the activator or downregulate the inhibitor. All right. So if there are no questions, I will switch now to the last part, which is that branched actin does more than just controlling cell migration. It also controls proliferation. Actually, this is the intermediate case I was hiding in the previous slide. Here, the carcinoma is still in situ. It means it's still contained within the myoepithelial layer. You've seen this expansion of this keratin 8 positive luminal population, which is really filling now the lumen of the gland. And you see that this luminal cell population, also we have many cases where Arpin expression is lost, but you still have expression in the myoepithelial layer that surrounds it. And here we were a bit puzzled because at this point, cells are not yet motile, yet they have already lost Arpin expression. So we were wondering why. And actually at this point, we remembered that the major controller of migration and of lamellipodium formation were the same ones that triggers proliferation. So for example, growth factors, you know, those are MCF10A cells, which I will use throughout the talk now. Those are human mammary cells. They are just immortalized, but not transformed. In other words, they don't make tumors if you graft them in mice. And if you starve them of growth factors, I will play the movie now, nothing happens. You see, the control population in full medium make beautiful lamellipodia, they translocate, they move somewhere, but these ones are just absolutely immobile. And of course, growth factors, serum, EGF, they're also notoriously involved in proliferation, and their name, growth factors. And so what we will use here is EDU. EDU is a small molecule which looks like thymidine, so which is incorporated in the cells when they are in S phase. So if you starve cells from growth factors, they do not proliferate, you have no staining with EDU. If you stimulate them with growth factors, they cycle. And when they cycle, they incorporate EDU during S phase, all right? So this is true with growth factors, but this is also true in wound healing. You all know that when cells reach confluences, they stop migrating, they also stop proliferating. And one way to resume both is to actually scratch, to wound the monolayer. So just to show you the migration, you induce a wound, you will see lamellipodia developing on two sides, and these cells crawl toward each other and eventually will close the wound. The same is true for proliferation. You see right next to the wound, many cells that were mostly stopped from cycling, stopped in the cell cycle, resume cycling, as you can see from EDU staining next to the wound. So, and actually it makes sense that the two are coupled, because if you want to repair a wound, it makes sense to multiply the number of cells and to make them move toward each other. So these two events collaborate to repair the wound. But it also raises the question of whether the same pathway, the RAC wave up-to-3 pathway, might be involved in cell cycle progression. And this was a topic I gave to this PhD student, Nicolas Molinier. And we started with simple experiment, just applying a small molecule which inhibits the up-to-3 complex, CK666, or structurally related but inactive control to MCF10A. And sure enough, you can block completely cycling if you block the up-to-3. This is true in this immortalized cell line, in many other immortalized cell lines, but this is true as well in primary mammary epithelial cells that we can get from cosmetic surgery. And these cells are absolutely normal and they are extremely sensitive to up-to-3 inhibition. And so this arrest is in G1 phase of the cell cycle. It's reversible, it's no apoptosis. If you wash away the inhibitor, cells will resume cycling. And so, of course, the obvious question is, which one of these branched actin is responsible for this control of cell cycle? And guess what? Sure enough, this is a wave. This is the one that controls lamellipodium protrusions that is responsible for proliferation. So you block proliferation if you remove wave. If you remove the inhibitor, Arpin, you have the opposite. So if cells are really cycling at full speed, you don't see any effect when you remove Arpin. But if you start to inhibit with CK666 in a dose-dependent manner, you see that you need a higher dose in Arpin-depleted cells to achieve a given level of inhibition. or yeah, so you need a higher dose of CK666, which makes sense since Arpin depleted cells make more branched actin. Same is true with RAC1. If you block RAC1 activation, you block cells, and that was known from before. And you can do the opposite. We have a genome engineered cell in MCF10A cell line, where one of the two alleles of RAC1 is activated thanks to this mutation, which blocks GTPase activity. And these cells have more protrusive activity. They are more persistent in migration. And here again, just like Arpin, you need more of the CK666 to achieve a certain level of inhibition. All right, so far so good. So I will cut a lot of data here because we don't have time, but basically using these two criteria, we could define a set of proteins that are absolutely required to give this proliferating signal. So you have RAC, wave, ARP23, coronin 1b, which detects somehow the branches. And we played with a different subunit of the wave complex. Most of them are required. For the ARP23, it was extremely sensitive for one of the two paralog ARPC1. ARPC1b was required and ARPC1a was not. And guess what? When we studied, we found that ARPC1b containing ARP23 is critical to form cortical branched actin, whereas ARPC1a containing was more critical to form branched actin at the surface of endosomes. And so we have a specificity here for some ARP23 complexes that, that are constituent of the cortical branched actin and that give this signaling pathway. All the data are there. And the real question at this point, which is interesting to ask, is why on earth would cortical branched actin control cell cycle progression? What kind of meaning does it have? Why would Mother Nature choose to monitor the amount of branched actin at the cortex? Well, you know that to regulate proliferation, you have three main parameters. The amount of growth factors, the fact that cells are well adhered to the extracellular matrix. They need to be adhered, but this matrix has to be sufficiently stiff so that the cells spread on it. And you know that cells should stop when they reach a certain density. This is a normal behavior. So somehow, and we don't really know how, cells sense density through the cell-cell junction, but it's not only having a free edge that controls proliferation. So you have these three parameters that are, we know, monitored, and that will help the cell to make its informed decision as to whether it will start a new cell cycle or not. Once the cell has decided to go for a cell cycle, it's blocked in G1, it will enter into S phase, and once it has entered into S phase, it will go through the whole cell cycle. So it's a decision, will I go or not go? But if you go, you make the whole cell cycle, and so one cells give two. And so this is a binary decision. It's an all or none decision. You go or you don't go. And again, to commit cells into a new state, we know positive feedback loops are very good because once you have triggered a process, you have an explosive amplification. So I showed you on this scheme that these components were required for the transmission. So it means in the best possible conditions, if you remove these components, cells will stop. So the best possible conditions, meaning full of growth factors, cells were adhered, and cell cultures being sparse, not too confluent, otherwise cells don't proliferate. If you have all these conditions, cells should proliferate. And if you block RAC wave or ARP23, cells will stop in the best conditions. So this is good to dissect what happens downstream of actin polymerization, but it's not good to understand what is upstream. And to go what is upstream, you should not block completely. You should rather sensitize the background. So what we will do here is the opposite experiment. We will make more branched actin, either by using active RAC, by removing the inhibitor, or by overexpressing ARPC1b. Always these three perturbations. And we will examine in which cases does it make a difference to have more branched actin. This is how we can realize in which conditions this pathway is important. So MCF10A are well behaved. Those are the black curves. If you give them full growth factors defined in laboratory conditions, they go full speed. If you starve them, they stop completely. If you increase the amount of branched actin by either one of these means, you see that basically it doesn't make much difference when you starve cells or fully stimulate them, but it makes a difference at intermediate concentrations, okay? So if you make more branched actin, you have an increase of proliferation in conditions where the stimuli is suboptimal. The same is true for adhesion. So here we played on the stiffness of the substrates, just because it's easier than to compare suspended cells to adherent cells. So we use polyacrylamide gels, more or less cross-linked, to coat, to present the fibronectin to the cells. And so they're exposed to the same amount of fibronectin, but in one case, this is stiff. And so we could continue this curve over here, it would go over here, or we can make it soft. And we can continue the curve over here. Here, what I'm showing you is a transition from soft to stiff. Morphologically, it translates into this kind of morphology. A stiff substrate allows cells to make protrusions and to spread. When the substrate is soft, here, cells remain rounded. You cannot see clear protrusions. The substrate is too compliant. When they pull on the substrate, they can grab the substrate, but you have some impurities. You can see that they are pulling on the substrate, but they cannot emit a protrusion. And in this case, they proliferate much less. So let's look now at our 3 cell lines where we make more branched actin. With active RAC, no Arpin, or more ARPC1B containing ARP2/3 complexes. Again, if the substrate is stiff enough, it doesn't make much of a difference. All the difference we see are when the substrate is soft. So in other words, if you make more branched actin, cells will cycle even though they should not, because the substrate is too soft, okay? So perhaps you're not very impressed by these differences, but what you have to realize is that one cells in our body, one of our breast cells, live in one given condition for all parameters. It is stimulated by a certain amount of growth factors. It lives in a soft environment. The breast is a soft environment. So only in laboratory we can explore these parameters of growth factors and stiffness, but there's no reason to explore them one by one. As when we explore them one by one, I showed you that we need in the control case to have a large amount of growth factors and a stiff substrate for proliferation. And it makes no difference if you remove Arpin. But as you can see, you have an extension of the hot colors in this phase diagram here when we remove Arpin. In other words, if the cell you consider lives somewhere here, when the stiffness is average, and this corresponds to the stiffness of the breast, and growth factor stimulation is not the maximum you can achieve in laboratory, but here, then you go from deep blue to hot color. So this is what's illustrated here. With full stimulation on both parameters, Arpin removal makes no difference. If one parameter is suboptimal, you have an effect, but which is perhaps not dramatic, but you have a 10-fold difference in EDU incorporation. So 10-fold difference in cycling, this is very significant if the two are suboptimal. So in the real life, branched actin is supposed to play an essential role. So we have the exact same result with RAQL and ARPC1B. I'm not showing you this. And so it seems that cortical branched actin, like the same one that produces lamellipodia when it can, and we know that lamellipodia are triggered by growth factors when cells are adherent, you have branched actin, which is polymerized, and this cortical branched actin is being sensed by this pathway to have an influence on cell cycle progression. So this is why Mother Nature, if you wish, has selected cortical branch tactin as a signal to decide whether to go for a new cell cycle or not, because cortical branch tactin seems to be an integrator of different inputs, okay? And there's one I didn't show you yet, which is a density-dependent effect. So again, black curve, regular MCF10A. MCF10As are well-behaved. When cells are sparse, they do proliferate. When cells are confluent, they stop from proliferating. This is the end of the black curve. If you make more branch tactin with RAGQL, or when you remove Arpin or ARPC1B, you have a little bit more proliferation at intermediate densities. In the case of Arpin removal and ARPC1B, haha, you have no effect for RAGQL. We don't know why. But clearly, cells are able to sense density at least through these two means. Now we did another experiment, which is to take confluent cells when they are stopped. And mechanically, we stretched these cells which are spread on PDMS extensible substrate, and look at the black curve. Over time, when you stretch sufficiently the monolayer so as not to break apart the monolayer, but just cells feel they have space, okay? Then some cells will resume cycling over time and start new run of cell cycle, just enough to occupy the space that the sense is free. It's a bit mysterious, but it works. And if you make more branch tactin with all three stimuli in this case, you see that they enter into this new cell cycle faster and more efficiently. More cells start a new cell cycle. So just to sum up, we have found that branch tactin makes a difference in response to all of these upstream stimuli: growth factors, substrate adhesion, and cell density. So it means those are the three categories of stimuli that control cell proliferation. So it means branch tactin is critical for all three parameters of controls over proliferation. Now let's look at cancer cells. So you know cancer cells are characterized by the fact they do not care so much about the amount of growth factors for their proliferation. They do not care so much about being attached to extracellular matrix. They will proliferate anyway. And that even when they reached high cell densities, they will continue to proliferate and form transformation foci. So we have some derivative of MCF10A which are transformed, which are cancerous. This is a derivative transformed by active RAS. It's called DCIS because it formed ductal carcinoma in situ. And let's look at their characteristic along these three parameters. They have, these cancer cells now have the exact same parameters as I showed you when you have more branch tactin. It means without growth factors, they stop. With full growth factors, they go full speed, as normal cells would do. But actually, to be transformed, render them more able to proliferate in conditions where they should not. And so we were really struck by the similarity of when we played with the level of branch tactin in our immortal cells, MCF10A. Then we decided to screen cell lines. So we have a lot of breast cell lines, and some of them do respond to CK666 by stopping cell cycle. Some of them do not at all. What was striking is that all cells that do stop were immortalized but not transformed, whereas all transformed cells were completely insensitive to CK666. So it means that the ability to proliferate despite the lack of branch tactin is a diagnostic, is a very good diagnostic of whether your cells will form tumors or not. And actually, we can convert MCF10A, which stops cycling upon ARP2/3 inhibition, into an insensitive cell line just by blocking well-known tumor suppressor genes, which are well known to control S-phase entry, the retinoblastoma protein or the cyclin CDK inhibitor. you see run the cell. If you deplete these two tumor suppressors, either of these tumor suppressors, cells are insensitive to CK666. So there are presumably many ways to run the cells insensitive to ARP2/3 inhibition, insensitive to the amount of branched actin. And this is where many of the known oncogenes are playing a role. So that would be downstream of branched actin, and that run the cells insensitive. But RAC itself is an oncogene. It's an oncogene which is mutated in melanoma patients. In 5 to 10% of melanoma patients, you have activating mutations of RAC1, which should make more branched actin, and then cells should still depend on this signaling for their proliferation. So we specifically collected cells harboring mutation on their RAC1. So you have a hotspot. This is this P29S mutations. Those are mostly melanoma, but we find it sometimes in breast cells or in sarcoma cells. And you can see, even though these cells are transformed, they're now sensitive to ARP2/3 inhibition because it's downstream. And so in this clonogenicity assay, all the cells that have an active RAC1 or RAC2 mutation are sensitive to ARP2/3 inhibition, whereas some other cell lines, the classical cell lines, which are not mutated specifically or RAC1, are insensitive. So to have understood this, give us a potential therapeutic option, right? But also we studied in quite detail the role of the ARP2/3 complex, the role of all its subunits. We understood this difference between C1b and C1a. And actually, we use the same cohort as I introduced you to already with Yvan Bièche to measure the expression of all genes by quantitative RT-PCR, all genes encoding ARP2/3 subunits. So the first striking result is that in the vast majority of cases, these genes are overexpressed and not underexpressed in patient tumors compared to normal tissues. And in many examples, this overexpression is associated with a poor prognosis for metastasis-free survival, as I showed you before. And of course, the most important, well, of course, this was what we expected, but we had to show it. I mean, the most overexpressed subunit is this ARPC1B subunit, which makes cortical branched actin, which is associated with a stronger effect in terms of prognosis, and ARPC1A is one of the least overexpressed. So just to show you the Kaplan-Meier, the metastasis-free survival, we have, with a single gene now, with ARPC1B, we have a very significant effect. So the P should be down here. No, or it will appear. Yeah, we have a very significant P that we had achieved only when we combined Arpin together with a significant subunit of the wave complex previously. So for sure, the RAC, the wave complex, and the ARP2/3 are very good prognosis factor to tell you if your cancer is going to be dreadful or not. This is due to two effects, to proliferation and migration in a coupled manner. Just because we are dealing with a multi-protein complex, we wanted to look at the biochemistry and not only the gene. So thanks to a collaboration with Yevgeny Denisov in Tomsk in Siberia, we got access to 13 breast tumors where we got the real tumors and we got the surrounding tissue, which is normal. And you can separate the surrounding normal tissue from the tumor, prepare extract, normalize the extract, as you can see here, with P150 glued, and check at the protein level the expression of ARP2/3 subunits. And if you do that on 13 couples, you see here in the brown, in the brownish tumors, that ARPC1B is overexpressed in 11 out of 13 tumors. And when you have an overexpression of ARPC1B, you also have an overexpression of ARPC2, which is a single subunit. So indeed, this is these ARPC1B containing ARP2/3 complexes which are overexpressed. So this is the end of it. Just to summarize, because you have a lot of information in this part, we found that branched actin, specifically the one at the cortex of the cell, which you can see in lamellipodia, but which exists even when you don't have lamellipodia at the cortex all around the cell, controls the G1S transition in normal cells. That is the decision to start a new cycle or not. The cortical branched actin is pretty good at doing this because it's an integrator. It will integrate all signals that control proliferation, the soluble ones from growth factors, but also the actin cytoskeleton, which generate forces, is very good at sensing forces and sensing forces from cell adhesions, either cell adhesions from attachment to the substratum or cell adhesions from engagement of cadherin making cell-cell junctions. Then we've seen that most of the driver mutations responsible for cancer development actually set cells free from the control of cortical branched actin. And this is how this is a very important pathway that will inform the cells that it's properly attached in the nice environment, that they should make one more round of cell cycle just to occupy the space in an harmonious manner. I mean, this signal is completely unnecessary, is misinterpreted by many driver mutations which occur downstream. But we've seen with the melanoma examples that if you have mutation in RAC1, which is upstream, then you can use ARP2/3 inhibition to block tumor cell proliferation, which can be useful for new therapies in some cases, new kinds of targeted therapies, because there's so far no therapies which were indicated for the patients having RAC1 mutations. And you've seen as well repeatedly in this talk how the deregulation of the ARP2/3 complex offers a diagnostic of cancer, and especially it's a prognosis for invasive cancers, because where you have more proliferation and more migration, of course, those are the most dreadful cancers. So with this, I would like to wrap up. I believe that I mentioned all the people who contributed this work I told you about in the lab. They're all gone, unfortunately, now. They have been replaced by a new gang of people who are contributing nice work, but this is for later. I think I mentioned the collaborators as well, and the funding, which is both for basic science and for cancer research. And I thank you for your attention. And we still have time for questions if you want, if you have some. So thank you.
Thank you very much, Alexis. This was fascinating as usual. It's always a treat to listen to one of your talks, and I hope they will flood you with questions. And if not, I will. So. Okay, please, some questions from the field. Yeah, Clara. Just open your microphone. You should be able to open your microphone. Yeah. Clara?
We cannot hear you, Clara.
I mean, I can see your microphone is open, but we cannot hear you.
Well, perhaps you can type the question in the chat and Miguel will read it.
Huh. Well, this is always a risk with these things, that sometimes the system works in its own way. So while Clara decides, or finds out how to ask a question, let me ask you something real quick. So there are a number of papers that also describe the signaling from cadherins to the ARP2/3 complex. And that's probably related to the ability of the cells to sense the level of packaging. I mean, what's going on there? I mean, is it the role of cryptic lamellipodia, or each cell has a different interprets? I mean, they branch out the cadherin signaling to interpret different things. I mean, what's your take on the E-cadherin to R2/3 complex signaling in this context?
So absolutely, yeah. Well, the difficult thing with adhesion is that adhesion to the substratum or adhesion to neighboring cells, you need the ARP2/3 to make the initial protrusion. And then after a while, you reorganize the actin cytoskeleton, and it's more pulling than actually, or either in stress fibers or adhesion belts, it's more pulling than protruding. And protruding seems to be a repair pathway. So you have an equilibrium between the pushing and the pulling in all these adhesions. And this is what you need for sensing forces. Indeed, the rack wave ARP2/3 pathway is critical for polymerizing branched actin at cell-cell junctions. This was demonstrated by results, I'm thinking, for example, of alpha-YAP.
Yep.
And cell-cell junction have a contribution for this density-dependent control over cell cycle progression, okay, which is often called contact inhibition for short, but it's an abuse of language, because contact inhibition, it's not as soon as cells touch each other that they will stop proliferating. They need to reach a certain density. So you have cells that are surrounded by neighboring cells with no free edge that have started to form cell-cell junctions, but that will still proliferate up to a certain level to reach a certain saturation density. And for example, with the NF2 tumor suppressor, which is also a component of cell-cell junction, what it does is just to modulate the number of cells. If you mutate this tumor suppressor gene, Merlin, encoded by NF2, you will just reach a higher level of cell density. So you have something which is still quite mysterious, which is this density sensing, which requires cell-cell junction formation, but not only. There's more to it. And indeed, the ability to form cryptic lamellipodia, as you say, is an important thing. For example, and those were results by the group of Giorgio Shita, who showed that you have jamming and jamming transition when cells reach confluence, epithelial cells reach confluence, they will stop proliferating, they will stop migrating, but at this cell density, you can resume both migration and proliferation just by single tricks, like adding a little bit of water in the medium that will play on osmolarity and that will change cell-cell junctions, and that will induce new rounds of proliferation, that will induce new lamellipodia beneath neighboring cells, this cryptic lamellipodia, and this will increase saturation density. And as I said, we have a whole program now to try to identify the molecular components of this saturation sensing, starting from the rack wave ARP2/3 pathway that we know pretty well of, to find new proteins that are critical for this monitoring of saturation density, and all of them, I can tell you in this screen, are involved in cancer progression, all of them.
Right, right. Clara, can we hear you now? No, apparently not. Well, while she's still fixing her microphone or whatever, because I mean, I think she's got all the permissions.